one particular part of this. We're going to kind of zoom in and look at the mitosis stage here is what we're going to check out. So we're doing this for, showing you pictures of it anyway, for one reason. We're going to do a lab on this that involves like onion cells, which is what's going to pop up here, where you guys are looking at the different stages and things like that of them. So if we start here, this is what most of your onion cells should look like here. And you remember that this is like these dark things, cell wall, cell membrane. What's that big dark thing in the middle? All right, and you notice this cell doesn't have one in it. Why wouldn't a cell have a nucleus in it? It's nothing more than this. Like think about an egg, right? Or eggy or whatever. And if you think about an egg and you consider the yolk the nucleus, when you make a slide, you just cut like a super thin layer, like one cell thick. Would it be possible for you to cut through an egg and not hit the yolk? That's all that happened here. When they cut through this cell, they just didn't cut through the part that had the nucleus in it. So some cells you'll find that just don't look like they have one, and that's why. When they took the slice through there, it just wasn't part of it. So these are the normal-looking cells that you'll mostly see. And this is, like, if you were looking at it as part of the cell cycle, this would be like the interphase stage. Where the chromatin, which is what's going to turn into the chromosomes, this is kind of loosely spread out, looks kind of granular in there, like dotty or splotchy or whatever. And that's exactly what you'll see under your microscope tomorrow. Looking here. And you can see in the nucleus here, it's slightly different. You can see like squiggles now, for lack of a better term. I don't know what the other phrase you'd use to describe, the other word you'd use to describe it is. But those things are the chromosomes. So that is taking the DNA, the chromatin that's in there, and it's condensed it into the very tight coils, and what you see there are chromosomes that are starting to form. So what you want down is this. As the cell is getting ready to divide, it kind of brings those chromosomes together and tightens them up. The first stage of this, where these things are all coiled up, is called prophase. And I'm not going to ask you for names or anything like that, but sometimes it helps to know, like, just to set order to it, if that makes sense. So the first stage, prophase, these things, the chromosomes are kind of coming together and tightening up like that. The next stage that comes after that, and a little bit different stain on this slide, but you can still see it pretty well, is all those chromosomes line up across the middle of the cell right there. So that's the very next stage, is that these chromosomes kind of get pulled into the very middle of the cell, and they line up across the middle. And what's called the metaphase plate is the name of that thing. And this is the stage called metaphase. This is also the stage where the centrioles, which are, if you remember back to our organelles, they're little tiny organelles in the cell that grow connecting fibers out of them. Essentially, if you think about Mortal Kombat, they're like Scorpion. They go to the edges of the cells and they send out these little fibers that hook onto the centromere, the center point of the chromosome, and pull them apart. Is this one, and you can see it a little better here now, that these chromosomes, they were lined up in the middle here, but now they're being pulled to opposite ends. And the name for that one is anaphase. And the chromosomes are getting pulled to opposite ends of the cell by the centromeres or the centrioles, sorry. So remember, your DNA doubled at a certain point. When they're all lined up in the middle, you've got twice the amount of DNA you need to have. And what's happening here is it's pulling that DNA apart from each other, because you only need one set of DNA in one cell and one set of DNA in the other cell. So this thing is just about to pinch off and become two cells there. Right now it's one cell with twice the amount of DNA it needs, and in just a couple seconds, this thing's going to pinch itself in half and just become two cells.